Hi dear students, welcome back to one more session of genetics. In the previous session, we had mentioned about the DNA fingerprinting techniques and what are the factors which are responsible for the variations between human beings, mainly the microsatellites as well as the mini satellites, which mainly differ in the length of the repeating sequences which have been present in the case of individuals. Now, if you go to look into the DNA fingerprinting technique, we had come across that we mainly use many satellites or mini satellites as a factors responsible for DNA fingerprinting. But coming to the modern DNA profiling techniques, also known as the short tandem repeat analysis, we mainly use the microsatellites rather than the mini satellites. Now, what is the difference between these microsatellites and the mini satellites? In the case of microsatellites, they are being found to be very short tandem repeats. Okay, they're just about 2 to 5 base pair long, and sometimes up to 10 base pairs. But in the case of what? In the case of mini satellites, they will be found to be a little more larger. That is, they will be about 100 base pairs and all that. So the difference between microsatellite and mini satellite is there. And in the case of DNA profiling, we mainly use to the what? Microsatellites to find out the differences between of the DNA profiling is mainly being done using the microsatellites of individuals. And just like the mini satellites, the microsatellites are also being repeated several times, but the number of repetitions is also been found to be high, and you can see that the length of the sequence which has been found to be repeated here in the case of a microsatellite could vary up to 2 to 5 or even up to 10 base pairs. So let's see the procedure. If you go to look into the steps which are being needed for the DNA profiling, you extract DNA from whatever sample you have, let it be blood, hair, nails, whatever forensic sample or any sample what you have. You can take the DNA sample, and then the DNA is being used as a template for PCR based amplification. Now, what is the PCR based amplification, or how is it being carried out? This PCR-based amplification is usually being carried out using primers which are specific for these short tandem repeats or the what? The microsatellites. And each primer is being found to be fluorescently coded, fluorescently labeled one. So, after doing a PCR reaction, of course, we have already gone across to the principles of PCR and all that. I'm not going into the details of that. It's a photocopying machine. So, different primers are being used here, and each primer is coding for a separate short tandem repeat or a microsatellite. And each primer will be coded with, it will be labeled with a different, a fluorescently coded tip. Okay, it will be differently labeled. And after the PCR reaction, an electrophoresis reaction will be carried out and the DNA samples will get separated. And once the electrophoresis is being done, now you might think that this is being done in a gel, as in a agarose gel in your lab. It's not like that. It will be done in a capillary electrophoresis in which these this DNA samples will get separated, and it's also connected with a laser detecting system which will detect the change in these probes. The fluorescently labeled probes will be detected and you will get a electrophoretogram. So you get a different profile pattern over here. So based upon this DNA profiling, okay, now you should understand each and every STR is being found to be labeled differently labeled over here, and like that you get a DNA profile. And using this DNA profile, you can analyze different individuals. This DNA sample, this profile can be loaded onto a database, and so even in different countries they have national databases where DNA profiles of such individuals are being stored, and so you can compare whether the DNA profile is matching over here. So let me just explain the steps once more. Here the DNA is extracted from the biological sample, and it is an incredibly sensitive method because only tiny amounts of DNA is being needed over here. And once what happens, the DNA sample is being used, you can carry out the profiling. Now, what sample can be used? It can be any biological sample, blood, saliva, hair, nails, etc. Different things can be used. And unlike the original DNA fingerprinting technique here, DNA profiling is not using restriction enzymes to cut the DNA, but instead the PCR or the polymerase chain reaction is being done. You can have a look at this. This is a case of DNA profiling where you take PCR with labeled primers. And if you want to just recollect what is DNA fingerprinting, you can have a look here. This was DNA fingerprinting where you are restricting, you are taking DNA and restriction, digesting and doing. This was DNA fingerprinting. Now we are talking about DNA profiling over here, and in DNA profiling we do not restrict digest the DNA, but you take the DNA sample and you do a PCR reaction. Okay. Now, what is PCR? You all know it is a polymerase chain reaction. In PCR, small bits of DNA called the primers, they will bind to the complementary sequences of the DNA of interest and mark the starting point of copying for the DNA of interest. And in STR analysis, the primers used in the PCR are designated or designed to attach to either end of the STR sequence of interest. That is, a short tandem repeats the primers in such a way that the primer will go and bind on at the end of the STR sequence. Now, and this primer is in such a way that it is also being labeled with a colored fluorescent tag. And as a result, what happens? It becomes easier to identify and record the STR sequences after PCR. And once the copies of the sequences have been obtained, you carry out the electrophoresis and you separate them according to the size. And each fragment passes by a laser which causes the fragments with fluorescent tags to glow with a specific color. And this particular output is analyzed by a series of colored peaks. So you get a profile, and different color will show the indication of each STR sequence. Okay, and the more the STR sequences which we use for profiling, it becomes more accurate that to identify a particular person. Now this is an example of a DNA profile which has been obtained from two different. Now if this is a suspect's DNA, okay, so his DNA was being taken and it was PCR amplified for some STR loci, okay, and that is short tandem repeats primers, which again some short tandem repeats were being done, and that this is the suspect sample, and a PCR analysis was being done and the profile was being obtained of that man. And this is the evidence sample, the DNA which you get from the evidence. That DNA profile is also being constructed. This is the electrophoretogram of one, and this is the second one. And when you compare, you can see that the peaks are being found to be similar. So you can say that what, of course, the suspect is in fact was being present at the sign of or the site of that particular incident. Now, if you go to consider the FBI have identified. Now, I'll want to tell you what particular sequence should be used as a short tandem repeat. When we have analyzed different individuals, FBI have determined that 13 core STR loci, or loci means STR locations or sequences, should be routinely used for the identification of individuals in the US. And in the case of Interpol, they have identified 10 standard loci in UK as well as in Europe. So, depending upon different countries, the STR analysis is being carried out using different STR sequences. So, this based upon your country, the country where this is being carried out, the STR analysis is being carried out using primers against different STR loci. And in the case of India, we have 9 STR loci, which we use for the identification of different populations. Now, You can also use other STRs, like for forensic purposes, you can use a YSTR, which is solely present in the male Y chromosome. So, for example, if you get a particular DNA sample and you cannot identify it as a whether male or female or not, you can use this YSTR to identify whether it is a of a whether that DNA belongs to a male or not. And only one person in every 10 million will have STR, will have a particular 10 million will have a what particular STR profile. So the chance of two different people to having the same STR is being found to be very less. So you get a unique pattern or a very reliable method using DNA profiling to identify different individuals. So this is now how do, as I mentioned earlier, all this DNA profiles can be stored in a database. Now UK is having in 1995, they have established a national database, and most of the countries do have national databases like that. And once it has been fully developed, like it's better to, it would be easier to identify the criminals and all that. So let me just conclude with it. You extract the DNA. Of course, you have to quantitate the DNA before a PCR has to be done. You do the amplification using PCR. You separate it by electrophoresis. Then you get the DNA profile and you analyze it and interpret it and check whether the quality is correct or not. Now, how can this be used to solve crimes? Of course, it's quite evident if that particular sequence, if, as I mentioned in the previous one, if a particular suspect, in this case, if the suspect and the evidence contains the same DNA profile, we say that it is of the same individual, or the person or the suspect was being present at that particular crime. Now, linking blood relatives, that also can be done by DNA profiling, because we always get half of the DNA from our mother and half of the DNA from our father. So STRs are also being passed down from the parents to the children. So DNA profiling can be used also to confirm whether two people are related to one another, and it is most commonly used to provide evidence that someone is not or is not the biological parent of a child. And you can also use DNA profiling to identify victims of a crime or major disasters, or even help bring separated families back together. This all can be done by the STR analysis, or the, what is it called, DNA profiling. And it is having a high success rate also. Now you can look at this. Now this is an example showing how DNA profiling can be used to identify the father, mother and the child. Now if